Okay, folks, so I thought I would finish up the rest of this lecture on enzymes and reactions. So we used to only talk about enzymes in biology as being proteins, but now we know that enzymes can also be ribozymes, or they just have an RNA component to them. Some examples of these we'll find out later in the semester are things like transfer RNA or messenger RNA. Right, so some of those are enzymes used in essentially making proteins out of DNA. But the important thing is that enzymes are catalysts, right? And so they speed up a reaction or the rate at which a reaction occurs. Even if a reaction is spontaneous, right, that doesn't necessarily mean it's fast. And so the example I talked about in class is if you have something like C6H12O6, that's glucose. Or maybe a better example is sucrose, which is C12H24O12. Right? So if you put sucrose in your coffee or in your sweet tea or whatever, it takes it, when you consume it, it doesn't take it very long for you to turn that into energy, which in you would be ATP. Right? But you could leave sucrose out on a table and it would slowly break down into CO2 and H2O, just like it does in you, and also in the process it would release some energy too, just not in the form of ATP. So this would happen spontaneously if you just left it out on the table. Problem is it'd be very slow, and you don't have that kind of time. You need to turn that into energy relatively quickly after you consume it and not sit around and wait. And so the way we do that is by using enzymes to convert that into energy. Whoops. I'll turn that off. And so the way enzymes are able to do this is through activation energies, or lowering activation energies, right? And so it doesn't matter what kind of reaction you have. If you're going to go from reactants to products, then you need to put some energy in. So I could go from something like sucrose to CO2, maybe some H2O. I could do that a couple of ways. I could just add heat, right, and get it to break down. And actually, I could keep going. You could put it in a pan until eventually all you ended up with is carbon, and everything else is gone. The problem is that takes a lot of energy to do that, right? So that's one thing you can do, is you can just add some heat. What's going to happen is that sucrose, if you have an enzyme anyway, that sucrose is going to bind with an enzyme. It's going to give you some transition state, right? So whatever that enzyme is, it's going to be bound to sucrose for a brief period of time, and then it'll be turned into CO2 and H2O. But again, you can either use heat to do it, or you can use enzymes to do it, and enzymes just lower activation energies. As living organisms, this is the preferred way, right? Because you can't afford to heat your body up to the extreme temperatures it would take to break down sucrose without an enzyme present, right? So enzymes are essentially the way to go. And this is just showing you an example of that. So if we were to graph it all out, here they're showing you ATP and glucose. You could use anything really. So if I were to go back to my sucrose example, right, which is just table sugar. If I were going to take table sugar and turn it into something else, turn it into some products, I could do it by adding heat. The problem is, if I add heat, then I need all of this input energy to do that. Right? So I need this much energy, right, to turn that sucrose into its products. If I have something like, if I have an enzyme present, then all I need is this much energy. I don't know why that keeps popping up. I need this much energy now. And so it really lowers the amount of energy I have to put into the system in order to get, and notice that at the end, I still have, right, so notice down here, I still have the same products, right, so nothing has changed as far as the end result. So I could still end up with CO2, H2O, and some ATP down here. And the other important thing is this enzyme is not used up in the process, right? So that enzyme remains. So that's another good thing about using them. You can recycle them over and over and over again. So the way these enzymes do this is, one, they will strain the bonds in the reactants. Maybe that yellow is a little tough to see. To make them eventually separate and turn into products. They might just position reactants together. So in the book's example, they're looking at glucose and ATP to give you what is essentially a glucose molecule, right, with a phosphate attached to it. Right? And so in order to do that, you could just take an enzyme, put glucose really close to ATP, and the reaction will take place. They may not be close enough just in the environment for that to happen. And then you can also change the local environment a little bit, maybe through some direct bonding of the enzyme with the substrate. Right? But typically, straining the bonds and the reactants and putting them really close together, that's enough to get reactions to take place. When we look at enzymes, some important kind of terminology are, one, active sites. So in class I talked about an enzyme called amylase. Right? Amylase is an enzyme that'll break starch down into sugar. And so the active site is where those two things, or where starch and amylase come together. So let's say amylase looks like this. Right? This is the same thing I drew on the board in class. Right? So if that's the enzyme structure for amylase, then, and I'll change the color here, then the structure of starch should look something like this. Right, so that should be starch. Right, so they should fit very closely together. Right? Again, I don't know why I drew that little extra line, but they should look just like that and go together, kind of lock and key. So where they go together is called the active site. Right, and so if these are the reactants or the substrate in which the enzyme acts on, so here starch would be the substrate of amylase. When you put those two things together, you get this enzyme substrate complex. Right? And that usually exists for a very short period of time, and then what's going to happen is, after these things go together very quickly, that starch is going to be broken down into simple sugars. And so this is showing you that essentially with glucose and ATP, right, to give you that glucose 6-phosphate structure. Again, that's just a glucose with a little bitty phosphate molecule stuck on it. Right? And so what happens is, here, this would be the active site of the enzyme. Right? You get your two substrates to come in and bind. The enzyme puts them really, really closely together here. Right? The reaction takes place, they split up, and you end up with your glucose 6-phosphate and your ADP. And then another important thing that they're showing you here is that when all this is done, right, this enzyme is just going to go back and start all over again. So the enzyme doesn't change. It can be used time and time and time again. And that's good for us because then we don't have to produce quite so many enzymes. We can make a little bit, and that'll do us. Right, so enzymes are super specific in what they do. They have a high specificity. Right? The amylase enzyme works on amylase and nothing else. You kind of have this lock and key metaphor, so enzymes work much like a key does in a lock. Keys don't work in very many locks, they work in very specific ones, and that's the same with enzymes. So amylase works on starch, nothing else. Some ways you can inhibit enzymes or stop them from functioning. One is through competition. Right? So if you just compete for the active site, so if I go back to something like the amylase that I drew, right? So if it looks like this, right, well, one way I could stop amylase from functioning is if I had some inhibitor that would bind here. So let's say, and I don't know why you would want to inhibit amylase specifically, but you could. If I had maybe some inhibitor molecule, right, that had a very kind of boxy structure that would fit into this part of the active site. Well, if it'll fit in there, that means it would stop the substrate, which would be starch, from fitting in. Right? So it's directly competing with this active site, so amylase could not function. Right? You can also have non-competitive inhibition, sometimes called allosteric inhibition. And so that's where you have some inhibitor that binds to something that's not the active site. And I can show you here. Right? So this is just up here is competitive inhibition. Right? So instead of the substrate binding, you have some inhibitor binding in the active site, right? And essentially shutting that active site down to the substrate. With non-competitive inhibition, what you could have is some inhibitor here binds to some allosteric site that has nothing to do with the active site. Right? But when you bind with that allosteric site, it changes the shape oftentimes of the active site and prevents the substrate here from binding to it. So non-competitive inhibition or allosteric inhibition. Within enzymes, you can also have things like, or these are just kind of various parts and pieces. So you can have what are called prosthetic groups, or cofactors, or coenzymes. And so all of these things are just things that may be needed in order for enzymes to function. Right? So maybe a cofactor for an enzyme might be copper. So in lab, we were looking at the use, or we were looking at enzymatic browning. One way you can stop enzymatic browning is through, it's called PTU, right? That's a compound, and it binds copper. And so if you sprinkle a little bit into solution, it'll bind all the copper, and the copper will prevent the enzyme in the potato from causing the brown coloration. And so in that sense, the copper would be a cofactor because the copper is needed in that particular enzyme to function. Right? So it's not just enough usually to have the enzyme alone. You have to have all the parts and pieces of the enzyme in order to make them function correctly. Enzymes have very specific temperature ranges, so this is showing the temperature range for humans, right? So about 37 C, which should be around 98.6 degrees Fahrenheit. Right? If you get off of that a little bit, then there certainly can be issues. Certainly if you go way high, right? If your body temperature gets way on up here, I mean 50 Celsius, super hot, you're dead, right, at that temperature. But if you notice how this curve is a little more slowly falling on the lower temperature side, and you can kind of see this in everyday life. So if someone falls into a frozen pond, right, and they lose consciousness and maybe even stop breathing, a lot of times their enzymes aren't severely damaged, and so once they heat back up, they're fine. If you go to Yellowstone, though, and you fall in to a hot spring, once your enzymes reach too high of a temperature, the term is denature. Right, so the enzymes denature, and so maybe an example of that is, again, if I go back to my amylase example, right, if amylase denatures, right, what would happen is essentially these active sites, why does that keep happening to me? These active sites would break down, right, and you could not get them to function again. If they get too cold, oftentimes they don't break down. Their reactions just slow down. If they get too hot, though, think about dropping and cracking an egg over a hot pan. Once it hits the hot pan and that the egg white turns white, that's denaturing the proteins in there, and you're not going to go back after that. Within these, so I should go back. So enzymes are used mostly in metabolic reactions or metabolism. There's other places they're used as well. And so when we talk about metabolism, we can talk about things that are catabolic or things that are anabolic. Right? And I usually talk to students or try to get them to see that anabolic things are things like anabolic steroids. So anabolic reactions are reactions that are endergonic. Right? And you go back to the other lecture for that. But basically you're building here. You're building with anabolic reactions. With catabolic reactions or exergonic reactions, you're breaking down things here. So maybe you're going from starch to sugar in a catabolic pathway, but in an anabolic pathway you might be going from sugar back to starch. Right? That would be anabolic. And so this is just showing you that how many reactions can be linked together with different enzymes. So I start off with an initial substrate and I have enzyme 1, put those two things together and I get intermediate 1, right? And then I have enzyme 2, put those together, I get intermediate 2, and then I need enzyme 3 before I can finally get my final product. And this is very characteristic of living systems. We are highly enzymatic organisms. We need a lot of enzymes to do every little tiny reaction before we end up with final products. And in the next few chapters, when we're talking about respiration, cellular respiration, and we're talking about photosynthesis, right? Photosynthesis, you'll see that. Right? You'll see how enzymatic everything really is. And you may wonder how we function at all, honestly, because everything is so enzymatic. So again, catabolic reactions, anabolic reactions. The point, well, one of the major drivers here oftentimes is ATP, and we need to make that in a couple ways. And really in the respiration chapter, cellular respiration chapter, we'll spend some time talking about this. But one way to make ATP is through substrate level phosphorylation. Right? That sounds super complicated, but essentially what that is the easiest way to make ATP, right? So remember what ATP is. ATP is adenosine or an adenine with 3 phosphates attached to it, right? Well, before I can get here, oftentimes I have something called a DP or adenosine diphosphate, which would be an adenine with 2 phosphates, right? Well, how do I get that other phosphate on there? I could just do something like substrate level phosphorylation. So for instance, if you know anything about the gym or people going to the gym, a lot of people take something called creatine. Right? Well, it's actually called creatine phosphate, right? So creatine with a phosphate attached. Creatine phosphate is said to help you recover more quickly, and we're talking seconds here, not huge amounts of time to recover. So you're not recovering much more quickly, just incrementally more, maybe. And so the way this works is with creatine phosphate, you just break the bond between the creatine and the phosphate molecule, and this phosphate comes down here and is attached onto ADP to get your ATP back. So that's substrate level phosphorylation. You've just exchanged phosphates between two things. In chemiosmosis, that's where you have, maybe you've got like a membrane with a bunch of positive charges on one side, a bunch of negative charges on the other, and so you use that concentration gradient, right, that electrochemical gradient, to help you do work. Right? So if you remember back to diffusion and osmosis, if I have a gradient, if I have a huge amount of one thing on one side of the membrane and something else on the other, well, these positive charges are going to want to go this way. Naturally, they want to go across. And so we can use that tendency, right? So if I put like a protein channel here, right, and pump those positives through that membrane, then I can turn that into work. I can make this do something for me. Right? Another way to think about that, if these positive charges are the water behind the dam, right? As the water flows through the dam, it's going to do work for me, and I can use that to make things like maybe ATP from ADP. Right? And so that's chemiosmosis, using an electrochemical gradient in order to make things like ATP. We'll talk a lot more about that in the respiration chapter. With these reactions, you've got oftentimes redox reactions, so oxidation and reduction. The easiest way to remember what oxidation and reduction reactions are is this little thing that was taught to me a long time ago, oil rig. Whoops, oil rig. Okay. So what this stands for is oxidation is loss, reduction is gain. Right? So what's that mean? Oxidation is loss. Well, in oxidation, you're removing an electron, so something loses an electron. In reduction, you get the addition of an electron. So oxidation is loss, reduction is gain of electrons. So if we look at this kind of generic chemical reaction, which one is oxidized, which one is reduced? Well, if you look here, right, from here to here, right, A is losing this electron over here. Right, it's gone. Right, so it's lost. So A is oxidized. Right, oxidation is loss. B is gaining that electron over here. Right, so reduction is gain. B is being reduced. Right, and so these reactions just kind of go together. If something is being oxidized, usually something has to be reduced in order for that to happen. Right, so those are just redox reactions. NADH is going to become hugely important. We need enzymes to make NADH, and we'll talk a lot more about it again in the respiration chapter, but it'll start out as NAD+, and then through a series of enzymatic reactions, we'll turn it in to NADH. And NADH is just a energy-carrying molecule that's going to help us make more ATP, and again, we'll focus on that more in the respiration chapter. And this is just showing you that the coupling there, that oxidation-reduction reaction. So how do we regulate all of this? How do we regulate enzymes in systems? Well, one, enzymes are proteins, and all proteins are coded for in the DNA. And usually when we talk about DNA, we can talk about genes, right? So if we can turn that gene on or turn that gene off, then we can regulate how much of that enzyme we have present. And depending on how much enzyme we have present depends on the rate at which we go through a certain metabolic process. Right? Maybe we also have some cell signaling going on with things like hormones. So growth hormone is a big one. So growth hormone causes all of your cells to grow, so you would be producing maybe a lot of particular enzymes to help you put things together. put molecules together in anabolic pathways or break them down in catabolic pathways, just through, again, kind of cellular regulation. And then one of the big ones too is through biochemical regulations, mostly through feedback loops, right, or feedback inhibition. Actually, feedback loops can either be positive or negative. Right? So, an example of, most of them in us anyway are negative. Most feedback loops are negative. Example of negative feedback loops are things like, if you think about your thermostat, right? So you set your thermostat on 70 degrees. And here in the wintertime, when your house gets colder than 70 degrees, the thermostat kicks the heat on, brings it up to temperature. Right? That's a negative feedback loop because you're changing the direction of the stimulus. A positive feedback loop is when you continue the direction of the stimulus. One of the classic examples of positive feedback loops in humans is childbirth, right? So as you get contractions, you get productions of oxytocin, which causes more contractions, which causes more oxytocin production, and that cycle just continues on and on and on and on and on. But the end point there is childbirth, right? So positive feedback loops have very definitive endpoints usually. Body temperature is an example of a negative feedback loop, right? So as you get hot, you start to sweat so that you can cool back down, right? You can see if body temperature was a positive feedback loop, as you get hot, you would just continue to get hotter, right? And so that's dangerous, right? So most of the ways we are regulated is through negative feedback loops. Right? So this is showing you kind of one way you can have a feedback inhibition or a negative feedback loop, right? So you have some substrate. Through some enzymatic reactions, you get the final product, right? And that final product can then come back and feed into an allosteric site in the original enzyme, right? And that's enough to kind of shut down this whole process, right? So once that binds, then this can't happen, and then the rest can't happen either. Right? And so that's essentially enzymes and reactions for this particular chapter. If you guys have any questions, if you would just shoot me an email, let me know. See you soon.